everybody, so today we're going to be focusing on the final macromolecule, and that is nucleic acids. So nucleic acids serve as an assembly instructions for all proteins in living organisms. And there's two different types of nucleic acids. The first one is deoxyribonucleic acid, or DNA, which stores the hereditary information that's responsible for inherited traits in all eukaryotes, prokaryotes, as well as viruses. We also have ribonucleic acid, or RNA, and this is the hereditary molecule of some viruses. There's also many different forms of RNA that are involved in protein synthesis in all cells. So an important concept, which is central dogma, is a two-step process by which information in our genes flows into proteins. So the way it works is DNA, which holds our genetic code and holds all the info that programs all cell activities, is made into RNA through a process called transcription. And RNA carries the message from the DNA to the cell, and it basically copies down all of the instructions from the DNA. And then the RNA is made into proteins through a process known as translation. And so an analogy to remember this is that DNA is basically that encyclopedia, which contains all of the mass information about everything. And we just want to know how to make a protein. So RNA is going to go in and look at how to make a protein, and it's going to photocopy that particular portion of the DNA or the encyclopedia. And then that photocopy is going to be used to make the protein. So we looked at nucleotides, which are basically, nucleic acids are the polymers of nucleotides, and the nucleotide consists of three parts that are linked together by covalent bonds. We have a nitrogenous base that's formed from rings of carbon and nitrogen atoms, a 5-carbon ring-shaped sugar, and 1 to 3 phosphate groups. Now, in a nucleotide, each nitrogenous base links covalently to a 5-carbon sugar, and this 5-carbon sugar can either be a deoxyribose sugar or a ribose sugar. And so, depending on whether we're looking at the RNA nucleotide or the DNA nucleotide, it could either be the deoxyribose sugar or the ribose sugar. And the sugars just differ in this chemical group at the 2' carbon. If that chemical group is an OH, it's going to be a ribose sugar, and if that chemical group is an H, it's going to be a deoxyribose sugar. And so, if it's a deoxyribose sugar, then it's a DNA nucleotide, and if it's a ribose sugar, it's an RNA nucleotide. So just the difference on that 2' carbon is going to determine whether it's an RNA nucleotide or a DNA nucleotide. Also, the nitrogenous bases will determine that, but we're going to get into that next. So the two general types of nitrogenous bases are pyrimidines and purines. Pyrimidines are bases that are single organic rings, and basically these are going to be uracil, cytosine, and thymine. Now cytosine is found in both RNA and DNA, but uracil is only found in RNA and thymine is only found in DNA. So like I was saying, that the nitrogenous bases as well as the 5-carbon sugar really determine whether it's an RNA nucleotide or a DNA nucleotide. The second type of nitrogenous base is the purines, and these are double-ringed or 2-ringed organic structures. So these are adenine and guanine, right? And both of these are found in RNA and DNA. So DNA and RNA consists of chains of nucleotides called polynucleotide chains, and each nucleotide is linked to one another with phosphodiester bonds. So the way this is done is if we look at the general backbone of the DNA molecule, we have this nucleotide attached to the next, and we can see that this attachment right over here is through that phosphodiester bond, which is basically each nucleotide is added onto the 3' end. So here is the 3' end of the nucleotide. So each following nucleotide is added to the 3' end of the previous nucleotide. And so a single bridging phosphate group between the 5' carbon of one sugar and the 3' carbon of the next sugar in line is going to be linked together, and that's going to form that phosphodiester bond. So we can see here we have the 3' carbon, and each subsequent nucleotide's 5' carbon is going to attach to that 3' carbon. And so if we have DNA, we know that the DNA molecule is double-stranded. So double-stranded means that each strand needs to link with the other strand, and that's going to be through the nitrogenous bases bonding with one another. So adenine is always going to bond to thymine with 2 hydrogen bonds, something like this, and guanine is always going to bind to cytosine with 3 hydrogen bonds, something like this. So you can see that since DNA is double-stranded, we're going to need the nitrogenous bases to bind with one another. And the reason uracil is not here is because uracil is an RNA, and RNA is not double-stranded, it's single-stranded, so therefore there's no base bonding that's going to occur in RNA. Also, in each base bonding, we can see an adenine, which is the pyrimidine, the purine is going to bind to a pyrimidine, and that is thymine. So here we can see a purine binding to a pyrimidine. So here we can see that DNA molecule coming together. So we have this phosphodiester bonds occurring in that phosphate-deoxyribose backbone, right? So we can see the 5' end is going to attach, the 5' end of this nucleotide is going to link onto the 3' end of this nucleotide, and side-by-side nucleotides are going to be joined or linked together through the nitrogenous bases, where we have the purines attaching to the pyrimidines with some number of hydrogen bonds, depending on whether it's A and T or C or G. And we can see that this strand is running 5' to 3' this way, and this strand is running 5' to 3' this way. So we can see that the strands are running in opposite directions, and this property is known as anti-parallel. So the DNA strands are always going to be anti-parallel, in which one strand is going to be running 5' to 3' this way, and the other one's going to be running 5' to 3' in the opposite direction. And subsequent nucleotides or additional nucleotides are always added to the 3' end. So in the case of this strand, each new nucleotide is going to be added to this end, whereas according to this strand, each new nucleotide is added to that end. So now we're going to look at DNA and RNA in comparison to one another. So DNA, deoxyribonucleic acids nucleotides, contain a deoxyribose sugar as well as a phosphate group and one of the 4 nitrogenous bases, adenine, thymine, guanine, or cytosine. Deoxyribonucleic acid is also double-stranded, right? And the 2 strands run in opposite directions or run anti-parallel to each other relative to the sugar phosphate backbone, and the bases on the opposite strands of the DNA hydrogen bond to one another to form that double-stranded DNA molecule. Whereas in RNA, its nucleotides contain the ribose sugar, a phosphate group, and one of the 4 bases, either adenine, uracil, guanine, or cytosine, and since it is single-stranded, it forms short linear forms as well as structures that fold back on themselves in clover or hairpin formations. So 2 nucleotides in particular that perform many functions in the cell in addition to serving as building blocks are going to be adenosine triphosphate and guanosine triphosphate, ATP and GTP. And they serve as primary molecules that transport chemical energy from one reaction system to another, as well as regulating cellular activity. And we can also see that ATP is the main energy currency in all living things. And we can see that ATP contains those 3 phosphate groups, hence the name triphosphate. And each time an additional phosphate group is removed, we have the release of energy. And here's the structure of GTP. But in general, both of these important nucleotides function in cells as building blocks or energy sources, providing energy, chemical energy to different reaction systems, as well as regulating cellular activity.